Hi everyone, today we are going to present an article about intricate regulatory circuit between FLI1 and GATA1, GATA2, LDB1, ERG dictates erythroid versus megakaryocytic differentiation. We are Isabella Santos and Sara Sanchez.
Okay, first of all, let's provide an introduction about some basic concepts that help to describe and understand the process of erythroid versus megakaryocytic cell differentiation mediated by transcription factors. Erythroid cells are primarily red blood cells, responsible for transporting oxygen throughout the body. On the other hand, megakaryocytic cells are the precursors to platelets and play a crucial role in blood clotting. So the interaction between transcription factors determines whether the progenitors will differentiate into red blood cells or into megakaryocytics. And we have the transcription factors. They are FLI1, which is responsible for inhibiting erythroid differentiation while promoting megakaryocytic differentiation by regulating the expression of GATA1 and GATA2, among other factors, and GATA1 that is essential for erythropoiesis and megakaryopoiesis. It is involved in the regulation of genes necessary or the development of red bloods and the megakaryocytics. And GATA2 is for transcription factors crucial for megakaryocytic differentiation. It's promoted by FLI1. LDB1 it interacts with various transcription factors, including FLI1 and GATA1. It plays a significant role in the regulation of gene expression during hematopoietic differentiation. And ERG, that is a transcription factor that negatively regulates GATA2 expression. Its overexpression can inhibit megakaryocytic differentiation.
Okay, here we can see the importance of FLI1 as it regulates expression of GATA1, GATA2, LDB1, and ERG. This expression helps us determine whether the stem cell will differentiate into an erythroid or megakaryocytic cell.
The general objective is evaluate an intricate regulatory circuit between FLI1 and GATA1, GATA2, LDB1, ERG dictates erythroid versus megakaryocytic differentiation.
Okay, methods. The first one is chromatin immunoprecipitation. This method is used to analyze the interaction between proteins and DNA in living cells. It allows for the identification of regions of DNA that are bound to specific proteins, such as transcription factors. The study suggests that FLI1 regulates the expression of GATA2 during the differentiation of hematopoietic progenitors. The second one is RT-qPCR. This method quantifies gene expression by measuring the amount of messenger RNA, mRNA in a sample. The results show that the decrease of FLI1 increased the expression of GATA1 and decreased that of GATA2, indicating a regulatory role of FLI1 in cell differentiation.
The third one is Western blotting. The Western blot is a technique that allows the detection and quantification of specific proteins in a sample through separation by electrophoresis and transfer to a membrane. The Western blot was used to confirm the expression of the proteins FLI1, GATA1, and GATA2 in HEL cells. This helped validate the results obtained by the RT-qPCR, providing a complete picture of protein regulating during differentiation. Flow symmetry is a technique that allows for the analysis of the physical and chemical characteristics of cells in suspension. It uses lasers to excite fluorochromes that label cellular components, enabling the quantification and classification of cells. It is used to study cellular heterogeneity and the expression of markers.
Okay, in electrophoresis, we will always have a GAF-DH that functions as a regulator in protein expression. It's a control that provides assurance. And okay, in this one, that is D, we can observe that if the SH-FLI1 station is inhibited, GATA2 is reduced compared to the other treatment, which is scramble. This can be seen in the role SH-FLI1 with GATA2, where it appears less colored, as we can see. Okay, now in this study conducted on 293 T cells, the expression of FLI1 is being reviewed. In the first graph, as we can see, it's not expressed, but in the second one, where they were treated, it is expressed, as we can see in this part. In this last graph, it's confirming that FLI1 directly binds to the GATA2 promoter, which we see by the reduction of immunoglobulin G. According to the results, this works to regulate expression of GATA2. Okay, B and D. In these results, a knockdown experiment was performed, which is a method that shows the inhibition of a gene, in this case the RNA of GATA1 and 2. In graph B, which was done with GATA, it's observed that both SH-GATA1 and SH-GATA1-2 almost completely inhibited gene expression, so the method worked. And in graph D, which was done with GATA2, SH-GATA2-1 and 2-2, almost completely inhibited the expression, but 2-3 didn't inhibit as much.
In B, we have that this shows that when GATA1 and 2 is silenced, there will be a decrease in GATA2 and LDB1, while if I1 will increase its expression slightly, which means that GATA1 is related to megacryoceric differentiation by regulating GATA2. So when GATA2 is inhibited, LDB1 decrease and FLI1 increase. And in the graph E, here they inhibit GATA2. So LDB1 decrease and FLI1 increase. In this graph, we can see that LDB1 genes were being silenced, and it was observed whether they continue to function or not. They still function, but at significantly reduced level. This was done to determine if a certain concentration of LDB1 is needed to block erythroid differentiation.
Okay, the first author is Liu F. et al. The association between FLI1 and GATA2 was previously observed, but it's not known whether the regulation is direct or indirect, so it supports the idea of the author. The second one is Ainomi P. et al., or expression of GATA2 in these cells induces megacryoceric differentiation and blocks erythroid mutation, so it was supported with the author. And the last one is Ben David et al. FLI1 homologue gene ERG is also implicated in both hematopoietic stem cell expansion and hematopoiesis, so it supports the idea of the author too.
In the conclusions, we have identified regulatory interactions between transcription factors like FLI1, GATA1, and GATA2, highlight the necessity of molecular biology in understanding cell differentiation processes, which is crucial for developing targeted therapies in hematological disease. In the second one, we have the techniques from molecular biology such as chromatin immunoprecipitation and RT-qPCR are essential for elucidating the complex mechanisms governing gene expression, providing insights that can lead to innovative medical treatments. And in the last one, we have identifying specific molecular targets involved in erythroid and megacryoceric differentiations opens new avenues for therapeutic interventions, demonstrating that the potential of molecular biology to address and treat various blood disorders effectively.